B3.1 Carry out a practical investigation in a biological context with guidance. So what does this mean? You must complete a whole investigation. Guidance means that your teacher will support you, but the whole process is student-driven. You will need to actively participate in the planning and recording of your own data. You will also need to keep a logbook of your draft method and the data you've collected. Your teacher may dictate which plant or animal you're going to investigate, but you'll need to choose the independent variable. Before you start, research your organism. What's its niche like? Where is it in the food chain? What abiotic factors affect it? How does your organism respond to changes in those abiotic factors? And what adaptations does it have to survive in that habitat? A statement of purpose is the aim of the investigation. What are you trying to find out? You'll also need to make a hypothesis, which is a prediction that can be tested. All of this will need to be linked to a scientific idea. So if you're investigating heart rate in Daphnia, for example, you might change the temperature of the water. You'll need to state how an increase in temperature may affect the heart rate and why. You need to complete a draft method for your teacher to sign off in your logbook. You may need to make changes to this as you actually carry out the investigation. One of the key parts of your investigation is the variables. The independent variable is the thing you're going to change. So, for example, in the Daphnia case, it might be the temperature of the water. A suitable range is within the organism's normal range, and you should have at least 4. The key thing is that you keep consistent with the independent and the dependent variable throughout the whole investigation. For accuracy of the dependent variable, think about how many times you need to repeat. When we're dealing with animals, it might not be as straightforward to get consistent results, so you may need to get between 5 and 10. And you need to think about which is the most accurate way of measuring the dependent variable. So, for example, trying to keep count of Daphnia heartbeat could be tricky. So how could you improve on this to actually make it accurate? Then lastly, the controlled variables. You need to think of ways of keeping things fair, and at least 3 for an achieved. Now, for merit, you need to have most. So think of all the other key factors that might affect your organism and make sure you keep those constant. So, for example, if you were changing temperature, you need to think about how you might keep light consistent. Now, any data that you collect, you must keep in your logbook. So that will be your raw data. When you put that into your report, you need to put it into a standalone table. What this means is that if I found just your results table, I should actually be able to work out what you actually investigated. Make sure the table has really clear headings with the independent and the dependent variables on there, including units. Also, calculate averages. To make the data usable, you can calculate the average then and identify a pattern. You don't have to, but you could make a graph of the averages, and it might be easier then to see the trends. Next, you need to interpret your data. So what do your results actually show you? Is there a trend? E.g., as the temperature increased, what happened to your dependent variable? It must link to the purpose, your aim. So if you plan to change temperature and see how it affected heart rate, that's what you must talk about in your conclusion. You must also have findings from another source. This could be another student's results who carried out a similar practical, or research that you found on the internet. For a merit, you must explain the biology behind your findings. So this is the how and why part. How does your organism respond in order to survive when you change an environmental factor? How is it adapted to survive both structurally and behaviourally? If you can link multiple biological ideas together, this then becomes a discussion for excellence. But don't forget to link those ideas to findings from other sources that you've found. For the evaluation, you can either write about the validity of your investigation or the reliability. For validity, you need to discuss how you made your results valid. This is about the controls you used. So why was each control important? What would have happened if you hadn't kept those things the same? Or you can discuss the reliability. How many repeats did you do? Were there any anomalies? How did you measure accurately? And lastly, don't forget to include a reference of any of the sources you used. When you hand in your report, you must also hand in your logbook.